This is how precision medicine uses your DNA to skip the trial and error of pill A and pill B and go straight to pill C, the right drug at the exact right dose. For decades, modern medicine treated us like standard population averages. But your genetics actually account for up to 95% of how you respond to a medication. So how does the exact same pill that perfectly cures your neighbor become absolutely toxic to you? Today, we are diving deep into your molecular lore to explain the crucial difference between pharmacogenetics, the sniper targeting single genes, and pharmacogenomics, the general scanning your entire system. Look at this grid. For the last century, this is exactly how the pharmaceutical industry saw you. Just another gray silhouette in a population average. They manufactured massive blockbuster drugs designed to treat the mathematical middle. But the Blockbuster era is dead. You are not a statistic. You are a complex molecular reality. And treating you like an average is exactly why your prescriptions are failing. Remember that sniper and general approach we just talked about? Here is what that actually looks like on your chart. When we use pharmacogenetics, the sniper, we are hunting for a mutation in just one specific gene to fix a single, isolated mechanism. But when we switch to pharmacogenomics, we zoom all the way out to analyze a massive, genome-wide web of polygenic interactions. It's the difference between fixing one faulty wire and mapping the entire electrical grid of your body. And understanding this grid is the only way to solve the 20 to 95% variance in your drug response. Remember that jump from pill A straight to pill C? Here is exactly how we bypass that lethal trial and error loop. For years, doctors guessed your dose using crude surrogate metrics, literally just measuring your body surface area like they were tailoring a suit. Precision pharmacology completely eliminates that guesswork. By analyzing your genetic profile first, we can actually predict adverse drug reactions, or ADRs, before the medication even touches your lips. The ultimate promise is absolute. The right drug for the right patient at the exact right dose. This brings us to pharmacokinetics, how your body actually impacts the drug. Think of your liver as a high-security sorting facility, controlling your metabolic clearance. If your genetics code you as an ultra-rapid metabolizer, the trapdoors are stuck wide open. The drug is flushed out before it does anything, guaranteeing therapeutic failure. But if you are a poor metabolizer, those doors are jammed shut. The chemicals pile up inside you until they trigger severe toxicity. This isn't theoretical. Testing for the TPMT enzyme actively prevents lethal bone marrow suppression when treating patients with the drug 6MP. And if a breast cancer patient lacks the CYP2D6 enzyme, giving them tamoxifen is completely useless because their liver literally cannot activate the drug. But what if your liver does its job perfectly and the drug levels in your blood are flawless? You could still be in massive danger. Welcome to Pharmacodynamics, the drug's actual impact on your body. Deep inside your cell membranes, drugs act like keys trying to unlock specific target receptors, like GPCRs. If you have a normal receptor, the key turns and the signal flows. But if you have a genetic variation, that lock is warped. The key just doesn't fit right. This results in completely altered sensitivity, changing how your cells respond through misaligned G-protein coupling or premature desensitization. To see exactly how this sabotage happens, let's pull up the ultimate molecular blueprint, the beta-2 adrenergic receptor, or ADRB2. This little architectural marvel is an intronless gene coding for a 413 amino acid GPCR. If we track its coordinates, it sits squarely on chromosome 5q31, a region heavily linked to asthma and IgE. Why does this specific blueprint matter? Because this single receptor is the primary therapeutic target for millions of people, relying on asthma inhalers and cardiovascular therapies. But this specific blueprint isn't just a static lock. It comes with built-in molecular switches. On one side, we have the Gly-16 switch. When this is activated, it heavily enhances downregulation. It physically commands the receptor to internalize, hiding inside the cell and turning the volume down on your medication. On the exact opposite side, we have the GLU27 switch. This is the stubborn roommate. It actively resists desensitization, forcing the receptor to stay awake and stay active on the surface. This brings us to one of the darkest paradoxes in modern medicine for patients who are Arg16 homozygotes. If you have this specific genetic makeup, taking a regular short-acting beta agonist, your standard rescue inhaler, gives you acute, immediate relief in the moment. But look at this graph. Over weeks of regular SABA use, that exact same inhaler triggers a steep, chronic decline in your lung function and a massive increase in asthma exacerbations. The very medicine designed to save your breath today is quietly stealing it away tomorrow. So, how on earth did massive clinical trials miss this LABA controversy? Why did some studies show extreme harm while others showed zero difference? The answer is the ultimate pharmacological cover-up: the steroid mask. You see, the ultimate confounder in these trials was that corticosteroids naturally force the body to upregulate or create more beta-2 receptors. When they co-administered these steroids during the trials, it literally patched over the underlying genetic defect. The steroids artificially balanced the scales, completely masking the massive genetic risk of the Arg16 mutation from the researchers. This massive clinical blind spot proves exactly why we cannot afford to just look at a single genetic letter. Single nucleotide polymorphisms, or SNPs, are not inherited one by one. They are inherited in linked blocks known as haplotypes. Because of a genetic phenomenon called linkage disequilibrium, that dangerous Arg16 mutation we just talked about is almost always physically co-inherited with the GLN27 switch. They are a package deal. By analyzing these haplotype pairs as a complete block, we can predict a patient's true drug response far better than if we just stared at a single genetic letter in a vacuum. But this molecular lore doesn't stop at your lungs. Enter biased agonism, the exact phenomenon controlling your heart and your pain pathways. When a drug ligand binds to a receptor, it doesn't just flip a single switch, it hits a biological fork in the road. Down the green G protein pathway lies the actual therapy and protection you want, but down the red beta-arrestin pathway lies the devastating side effects. By understanding this exact split, we look at the ADRB1 receptor to predict which cardiovascular patients will actually get a survival benefit from beta blockers. Even wilder, we use the OPRM1 receptor to surgically separate the sweet relief of analgesia from the absolute horrors of addiction and respiratory depression. We are standing at the absolute edge of a medical revolution. The future of pharmacology is moving away from just temporarily occupying a receptor to actively eliminating it. We are deploying PROTACs, molecular hitmen designed to completely destroy disease targets we previously thought were undruggable. We are moving beyond single genes into polygenic risk scores, using AI and multi-omics to integrate massive sets of DNA, RNA, and protein data. Your therapy is finally being tailored, not just to your disease, but to your exact, unique molecular biography. But knowing how genetics rewrite your receptors is only the final piece of the puzzle. To truly master the complete lore of pharmacodynamics, you need to understand the foundational drug-receptor interactions that make all of this possible. Click the playlist on your screen to complete your journey, and I'll see you in the lab.